Yes, ma'am. Am I audible?
Yes, yes, you are audible.
Okay. A very good morning to one and all. On behalf of Principal Kmkcp College of Pharmacy, I'm Melroy Disa, Assistant Professor in Pharmacy. Welcome you all to the fifth and concluding day of AICT-sponsored online faculty development program on emerging trends in drug engineering. I now request FDP Coordinator Dr. Mrinal Sanae to give us a gist about the sessions conducted yesterday and to provide us a brief overview about the proceedings for today. Over to you, ma'am.
Morning. Good morning. Thank you, Milroy sir. Good morning, Dr. Datar sir. And good morning, Dr. Mandar sir. Today is the last day of our faculty development program, and we are beginning with our sessions. I would like to make one announcement that there is a slight change in the schedule. Today's morning session will be conducted by Dr. Ajit Datar, and it will be, I'm sure it will be a very interesting session with such a senior person who is knowledgeable in the field of, who has a lot of knowledge in the field of LCMS is there with us, and I'm sure participants will really enjoy his session. To brief you about the yesterday's session, we had Dr. Bange, who delivered a very enlightening session on the green chemistry procedures, various process strategies which can be adapted for environmentally benign synthetic molecules, and to take into consideration the pollution through which we are undergoing. Such methods need to be adapted. Even there was a session by Mr. Pramod Dahibate, who updated very nice to us about nice information on the regulatory requirements, and especially US regulatory requirements for the investigational new drug. Then there was a session by Dr. Kiran Jathar, who also delivered a very interesting session, giving us an information about how green chemistry can be applied in the various sectors of the science. And now today we are having a very senior person in the field of analysis, and that is Dr. Ajit Datar. I request our faculty member, Dr. Mandar Malik, who is the assistant professor in Department of Pharmacognosy, to introduce to the participant. Over to you, sir.
Thank you, ma'am. Good morning, everybody. I welcome you all in the final day of this magnificent Atal FDP. I am grateful to the organizing committee and Dr. Mrinal Sanae ma'am for giving me this opportunity. I'm also delighted to introduce today's speaker, Dr. Ajit Datar sir. Dr. Datar is working as an advisor to the Borosil. Previously, he was working with the Shimadzu Analytical India Private Limited, Mumbai, in the customer support center. His main job profile is to provide instrumental analysis support to the customer and provide training as an advisor. Prior to Shimadzu, he was working with the Electron and served this organization as general manager to this division of advanced mass spectroscopy. He was responsible for promoting advanced mass spectroscopy products of Thermo Finnegan in India. He retired in 2007 after completing 58 years of his service. He served for Cromlin and IR Technology as technical director and senior manager respectively for a period of 15 years. His first assignment was with Central Research Station of Associate Cement Cost Limited, that is ACC Cement, as research officer. He served ACC for 12 years. He has developed method for characterization of catalyst and process product using catalyst. He indigenously developed DET surface area analyzer and particle size analyzer. Dr. Datar received his MSc in 1971 and PhD in 1976 from Mumbai University. He was recipient of Department of Atomic Energy Fellowship in 1971 and carried out his PhD research from Analytical Chemistry Division of BARC itself. He is currently also working adjunct professor in Khalsa College and visiting faculty at several other colleges in Mumbai. He had several publications in his name, and he also guided PhD students in Mumbai University. On the last final note, he also fascinating individual, and we grateful for his presence among us today. Without further ado, I request Dr. Datar to enlighten us with knowledge and experience in LCMS application for studies of metabolic identification. Over to you, sir.
Yeah, please stop
Good
share.
morning. Yeah, good morning, and thank you everyone. And thanks a lot for my introduction and kind words about me. Today, it's a real surprise for me that I had to come here and give a lecture within something like 1 hour's notice. Okay, so probably you may find me, I'm not really well arranged, but pardon me if there is some jumping from here and there within the presentation, but I would try to be as
So instead, we are highly obliged to have you in a time of a need, and without any hesitation, you were ready. So
my
we
pleasure,
are
of course.
thankful to you for
Yeah.
being with us and helping us for the smooth conduct of this FDP.
Yeah,
Thanks a lot.
My pleasure, ma'am, because I have been working with Kmkcp Kunnani College for last several years, right from Dr. Lala's days when he was principal of the college and when the college was in Worli. So I used to come to give lectures when the college was there, and I continued having my association with your college. Thanks to Dr. Purnima madam, because she is always associated and connected with me for whenever there is any requirement. She has always approached me, and because of her, I still have a connection with your college. Yeah. Okay.
We
So
are blessed to have her.
yeah. So let me try to talk to you about LCMS and its application in metabolite identification. So the actual talk could be concepts of LCMS and its application to metabolite identification, drug metabolite identification. So let me share my presentation, and we'll give me some time for that. My title for you is the concepts of LCMS and its application for metabolite identification. Okay. Fine. So let me begin, and I always would like to begin my presentation with a juggler who talks about analytical chemistry. Okay, so my next slide would be some of you are familiar with it because you had been probably listening to my presentation. Some of you are at least listening to my presentation for some time on various subjects. I probably had given a talk, but this slide always comes first, because I would like to introduce a friend of mine who is nothing but analytical chemist. Okay, and I am a pure and poor analytical chemist. So he is analytical chemist. He is playing with some important terms in analytical chemistry, and what those terms are: they are sensitivity, selectivity, separation, structure, and speed. I need not have to tell the importance of all these terms, as you are familiar with the analytical instruments and analytical separation techniques, as well as analytical identification techniques, etc. So these terms are quite known to you. But according to me, these are 5 important S of analytical chemistry. And to manage to play with these 5 S, you require to do some kind of a separation of some of the errors. And those are basically sampling error, background, and noise. These 3 things need to be suppressed so that one can play with the 5S very well. So to be a good analytical chemist, you have to have minimum sampling error. Sampling error has 2 parts: how you select the sample and how you prepare your sample. Okay, so both these are very critical. Background is always associated with the sample which is being analyzed, especially at very low level, because your matrix is huge compared to the analyte molecule which you are trying to measure. So the matrix will always give you a lot of background. And then matrix itself can give you some noise, plus there could be inherent noise which can come because of electronic components which are used in instrument. So noise is always associated with it. And if you reduce your noise, your signal to noise ratio will improve, and signal to noise ratio definitely talks about the sensitivity of your method. So these 3 things need to be suppressed so that one can play with the terms like sensitivity, selectivity, etc. properly. And especially when we talk about the analysis with respect to metabolite identification in biological matrix, it is very essential to control these kind of errors so that you will be able to detect a very small amount of metabolite when it gets formed, when you do some kind of in vitro or in vivo studies. Okay, so whether it is a BAB studies or whether it is a metabolite identification studies, you require to have the control on some of these parameters. So let me quickly go to the techniques of importance, especially as far as metabolite identification is concerned: GCMS, LCMS, and MSMS. Out of which, of course, LCMS is more important than a GCMS, because most of the compounds being non-volatile, GCMS has limited applications. Yeah, why we require to have mass spectrometry? So just quickly, I will go through that. Chromatography gives you only one dimension, that is retention time. But mass spectrometer gives you additional dimension, that is molecular weight, which is very important to identify the compound. And then it also gives you fragments of that molecule, which will again be very useful to find out the structure of a compound. So the LC-MSMS or GCMS give you all these kind of informations which help you in identification of a compound. I'm not going into history of GC, LC, and MS, but just very quickly, LC and MS started together somewhere in 1900. Okay, but then they could meet only after about 70 years. Okay, so till 70 years, LC was alone, MS was alone, and after 70 years of individual working, LC could meet MS, but it did not happen with GC. GC was invented in 1952 by James and Martin, and within 4 years, GCMS came into existence. Okay, so we have to understand why it is so, and we will quickly get into the next part, which will help you to understand what is the difference between these two techniques. So I will definitely cover some aspects of GCMS before we get into LCMS. So let us quickly go into the mass spectrometry as an instrument. So these are components of a mass spectrometer: sample inlet, ionization, mass analyzer, detector, and data system. Sample inlet could be GC or LC for us, as far as today's lecture is concerned. Then we have ionization. So sample coming from GC or LC gets ionized, gets vaporized also, and the ions enter into the mass analyzer in vapor form. They are separated by mass analyzer on the basis of mass to charge ratio. Then they go to the detector. Detector senses them and gives a signal which is handled by the data system. You require a very high vacuum of the order of 10 raised to -5 millimeter, so that the ions which are generated in a vapor form can be easily transferred to the mass analyzer, and through the mass analyzer to the detector. Okay, so the free movement of ions which are in vapor form, you require to have a high vacuum in this region. Okay, so these are all the components of a mass spectrometer. We have different methods of ionization. I'm not definitely going to cover the GC or LC, because those are our sample inlet points. After that, we need to ionize the sample. So ionization methods are very important. When we have volatile samples, which are generally analyzed by GC, we have two methods of ionization called as EI and CI. For non-volatile samples, and the thermally labile samples, we generally use LC for them, and we have several methods. Some of them are now outdated, and we use some of the modern methods like electrospray ionization, atmospheric pressure chemical ionization, atmospheric pressure photoionization, which in all are called as atmospheric pressure ionization. So these three techniques are part of atmospheric pressure ionization. They are used today with LC, and LCMS success has come because of the atmospheric pressure ionization technique. Of course, there is another technique when you can convert non-volatile into volatiles by derivatization, chemical derivatization. So the compound becomes volatile, and you can analyze them by GC or GCMS, and some compounds like fatty acids can be converted into their methyl esters, and they become volatile, and they can be analyzed by GCMS. Okay, so ionization is the most important thing in the mass spectrometry. I have been using 3 punchlines for a very long time, and those punchlines are: if you ask GC what you can do, GC will tell you that if you can evaporate, I can separate. If you ask LC what you can do, LC will tell you if you can dissolve, I can resolve. This is the term which is there in one of the HPLC book. And if you ask mass spectrometer what you can do, it will tell you that if you can ionize, I can recognize. So ionization is the most important thing in mass spectrometry. Okay, so this is about a GC. I'm not going to talk about it. We have technique which is called as electron ionization and chemical ionization. These are the two techniques which are used for GCMS. We'll quickly go through them. Electron ionization is highly energetic process. We bombard molecule with 70 electron volt energy of electrons, which is about 1600 kilocalories, and that's quite high. Because of that, you get a lot of fragment ions, and you may get a molecular ion, and sometimes you may not get a molecular ion. So that means the molecule is fragile, you may not get a molecular ion. If molecule is strong enough, you may get a molecular ion. So molecular ion gives you molecular weight. If you do not get a molecular ion, you will miss molecular weight. Okay. Now, one good advantage of GCMS is that as far as you use 70 electron volts as energy, you get a spectra which is every time repeatable. So if you do the compound today and if you do the compound after 1 year, you will get the same spectra if you use a GCMS with 70 electron volts. On any GCMS manufactured by anybody, the same compound will give exactly similar spectra, and that is why there was a library generated, and there are commercial libraries available which contain more than 3 lakhs of spectra stored in them. Okay, so library is one good advantage of GCMS, which help you in identification of any compounds. So it works like that. So you have electrons generated by heating the filament, noble metal filament, and they travel to the anode. Your sample comes from your GC, gets ionized by bombardment of electron. It loses electron from the external orbit, so molecule becomes positively charged, and these molecules which are positively charged, they are ions, and also the fragment of them, they enter into the mass analyzer, which is on this side. Okay, so that is how the ionization happens. This is how the, I mean, if ABC is a molecule, you can get ABC plus, which is your molecular ion. You can get AB plus, that's a fragment of ABC molecule. You can get BC plus, you can get AC plus, you can get A plus, B plus, C plus. So a simple molecule like ABC gives you fragments like this, and knowing the mass of each one of them, you can identify what ABC is. Let us look at one simple example. This is a compound known as acetophenol, and you can see these are my ABCs. Phenyl group, C double bond O group, and CH3 group. Okay, so A, B, and C. This is the molecular weight of the compound 120. It loses methyl group, so the bond gets broken here, it gets fragmented here, and you get AB plus, and this is your AB plus 105. Then this is 77. Sorry for the mistake, that 77 has moved here, number has moved here, but this is your 77, which is phenyl group, so you are getting A plus. 43 your BC plus. Okay, so COCH3 molecular weight is 43. So you are getting information like this about this molecule, and when you look at the library, it will definitely predict the structure of the compound as acetophenol. Okay, when it sees this kind of a spectra. Now what is important here is the position on M by Z axis. This is M by Z axis, mass axis, and also the intensity of each peak. So what is done is the maximum peak, the maximum intense peak is considered to be 100, and then with respect to that, the intensity of all other peaks are calculated. So this ratio, that is 105 to 120, 105 to 77, 105 to 43, all these ratios are also equally important compared to the position of these peaks. So when everything matches, then only this compound is identified as acetophenol. Okay, so that is how the library works and gives you the possibilities of the compounds which are existing in your sample. And then it will also give you, because it may give you some 4 or 5 possibilities which are very close to the structure of this. Okay. And the first one, which will have a maximum score, so it also gives a score. So first one will have maximum score, and generally that could be your compound. Not necessarily, but that could be your compound. Okay, so that is how it works. Now, as I said, sometimes you may not get a molecular ion. And in such a case, especially when you work with some unknown kind of a compound and which may not be there in library, and then you are not sure whether the spectra given by Gcl Cnms is right or wrong. So you can use a method which is called as chemical ionization, which will definitely give you molecular weight information. Okay, so it definitely gives a molecular weight information, not directly, but it gives you a protonated molecule, M plus H gets formed, and with that you get a molecular weight information. Only what you have to do is you have to use some kind of a reagent gas in the electron ionization chamber, and it could be methane, isobutane, or ammonia. Okay, one of these three are used as the reagent gases. As there is a variation like this, the fragmentation which may happen a little bit, it will not be uniform with each of these gases. Even the pressure of gas can change the fragmentation. That is why there is no CI library. Okay. So this is how it works. So this is the same ionization chamber. Now the methane gas is introduced. It gets ionized. The sample comes from the GC. It gets ionized and then enters into the mass analyzer, which is on this side. So CI gives you molecular weight information. EI may not give you molecular weight information. Let us go into the example straight away. So this is the EI spectrum of amitriptyline and cyclobenzapine. So you will see that their molecular weights are 277 and 275 respectively, and there is no molecular ion seen. 277 is missing, 275 is missing in EI mode. But once you go to CI mode, positive ion chemical ionization mode, you can see the molecular weight is 277, proton added, so molecular weight is showing as 278. And so you know that if you subtract one, you are going to get a molecular weight. Okay, and you can see the intensity of this ion. It is very strong ion, so you'll never miss it. So molecular weight information is definitely available to you because of CI. Okay, so that is the advantage of a chemical ionization, and also EI, because EI gives you fragment by which you can get the library search possible, and you can get the structural information by using a library. This is how GCMS looks like. So this portion is GC and this is MS. So MS is very small compared to GC. Okay, so just to give you some information about how the instrument looks like. We'll quickly get into HPLC. Okay, so in volatiles, HPLC generally we use for in volatiles and thermally labile compounds. So when we have thermally labile compounds and in volatiles, it is very difficult to put them into vapor phase because mass analyzer require a compound in a vapor phase and also charge. It should be ionized. And to convert the in volatiles into vapor phase or thermally labile compound into vapor phase is a challenge because they will decompose before their boiling points. Okay, so the techniques, the combination of LCMS was felt to be impossible initially. So people tried to develop some techniques like field desorption, fast atom bombardment, secondary ion mass spectrometry, thermospray. This was quite popular initially as LCMS interface, but all these could not give the kind of a sensitivity which was possible by GCMS. So people felt that LCMS is really a challenge and it may not be very successful to have LCMS at all. Okay, so this is what is going to happen in case of LCMS. That is what felt, that this love affair between two species, one which is in a vapor phase, one which is in a liquid phase, they cannot come together, and this love affair is impossible. In addition to that, LC mobile phase generates too much amount of gas to be handled by vacuum system. Now, in case of a GCMS, we use 1 ml per minute of a carrier gas flow rate. In case of LCMS, we use 1 ml per minute of a mobile phase like methanol or water. Now, if you convert 1 ml of a liquid into vapor phase, it would generate too much amount of a gas. I can just work out a very quick calculation. So let us assume that we are using entire aqueous mobile phase, 1 ml of water as a mobile phase. Now 1 ml of water would generate 1.2 liters of a gas, 1200 ml of a gas at NTP. while in case of a GC, capillary column GC, we are using 1 ml of a gas as a mobile phase. So that much amount of gas can be easily handled by vacuum system, but 1200 ml of liquid converted into vapor cannot be handled by a vacuum system. 1 ml of liquid will get converted to 1200 ml of vapor. That cannot be handled by a vacuum system. Okay, because this is based on the calculation that 1 gram mole of any substance gives you 22.4 liters of a gas at NTP. So 1 gram of water, which is equivalent to 1 ml of water, will give you 1.2 liters of a gas. Okay, so that is why it was felt that the vacuum system cannot handle such a huge amount of a vapor which enters into the vacuum system. Plus, the compounds which are to be vaporized, they get decomposed at high temperature. So to make them into vapor phase is difficult. Okay, so that was felt that it was impossible to have a LCMS, but then the old techniques slowly became obsolete and new techniques evolved, like electrospray, atmospheric pressure, chemical ionization, etc. Matrix assisted laser desorption, which is used as an independent ionization source. It is not used with LC, but these two along with atmospheric pressure photoionization are used with LC. Okay, so I will just quickly go through this thermospray interface just to indicate to you how this used to work. Okay, so here is the nebulized mobile phase coming out in form of a spray through the heated interface here. So this is heated, so everything gets vaporized, and then you have EI filament. So you ionize it by again electron ionization, so you get a lot of fragments, and you have generated 1.2 liters of a gas along with your sample molecules. So some sample molecules may enter into the mass spectrometer, but while most of the mobile phase will go here, that is what is expected, but a lot of mobile phase vapors also will get into mass spectrometer, will give you huge background, and will create some problems for the vacuum system of a mass analyzer. And because of that, this kind of a interface never worked with very high sensitivity. So we had two great people who introduced two different interfaces for involatile compounds or non-volatile compounds. John Fenn, who introduced electrospray ionization, and Koichi Tanaka, who introduced MALDI, almost at the same time, and that is why both of them received a Nobel Prize in the same year 2002. One got it for electrospray ionization development, one got it for MALDI ionization development. Okay, so both of them really made a lot of changes in the understanding of non-volatiles by LCMS. So atmospheric pressure ionization is the current way of ionizing the molecules in LCMS, and the major difference is that we are not introducing sample into the vacuum, we are ionizing the sample at the atmospheric pressure. Okay, and there are three techniques: electrospray, atmospheric pressure chemical ionization, atmospheric pressure photoionization. All are soft ionization techniques, give only molecular ion and not the fragments. Okay, so this is how electrospray works. So you have nebulization happening, so you form the spray, small droplets of your mobile phase are created. Only when they pass through this capillary, it is called as electrospray capillary, it is given a very high positive potential, because of which the droplets would be positively charged. This is in atmospheric pressure. There is a high flow rate of a nitrogen gas in this area, and because of the flow of nitrogen gas and because of some temperature applied to the capillary, the drops will start evaporating. And as the drops evaporate, the positive charges start coming closer together, and then there will be repulsion starts. One is the Coulombic repulsive forces of the positive charges, and other is the surface tension force within the drop, which will try to keep the drop intact, and the repulsive forces will try to go away from each other. And finally, when the drop shrinks further and further because of the evaporation of the mobile phase from the surface, the repulsive forces become more stronger than the surface tension force, and drop shatters, breaks into small droplets, and finally the charge will get transferred to the molecule, which is solvated with the mobile phase. So molecule of analyte never sits alone. It is always solvated, and as the drop evaporates, the charge gets transferred to the molecule. Now nothing is holding it, so it comes into vapor phase, and as it is charged now, it enters into the mass analyzer, and that is how the electrospray ionization works. So as I said, it is the theory. So drop becomes smaller and smaller, it breaks, and finally you get ions produced which enter into the mass analyzer. Okay, yeah, so this is how the spray happens. So these are all solvent molecules, mobile phase molecules. Ions which are formed enter into the mass analyzer. Okay. So electrospray is the atmospheric pressure ion source. Smaller molecules may be singly charged. High molecular weight samples become multiply charged like proteins, and molecular weights up to 150,000 Daltons can be measured accurately. So not only the small pharmaceutical molecules, it can be used for peptide-based pharmaceutical compounds, it can be used for protein-based pharmaceutical compounds, so LCMS can be used for all types of compounds. Yeah, so there is another one which is called as atmospheric pressure chemical ionization, which is used for small molecules. It is not used for large molecules like electrospray, but it is used for semipolar compounds. Okay, so like steroids are semipolar, lipids are semipolar. So it is used for small pharmaceutical compounds and steroids, etc. Okay, so I'm not going into details of that because I want to cover some aspects of application as far as metabolite identification is concerned. So let me quickly go through the theoretical aspects. So we'll just have a quick comparison of various techniques. EI, which can be Cms, can handle non-polar molecules up to mass range of 800 maximum. Fab, which I have not covered, can go up to 5,000 molecular weight and cover a lot of polar compounds. Like it was the first one which could handle sugar molecules or peptide molecules. So Fab was very useful for biological research in the earlier days. Okay. Then thermospray, which is used for semipolar molecules and goes up to 1200 molecular weight. Then we have atmospheric pressure chemical ionization, which is used for semipolar molecules, goes up to 1000 molecular weight. Then we had APPI, which I have not covered, atmospheric pressure photoionization, which is used for non-polar molecules up to 600 molecular weight. And then we have electrospray ionization, which goes up to 150,000 molecular weight and covers a lot of highly polar compounds as well as some semi-polar compounds in some areas. And you can see these three are atmospheric pressure ionization techniques. Because of these three techniques, the LCMS has become possible now, and the atmosphere has made the change. And that is why I put another cartoon. I drew another cartoon which says that the atmosphere has made the change. The cock in the previous picture has compromised. He said, I can marry duck, which can also stay in water as well as in atmosphere. The marriage is successful. There is a chickling. It doesn't say quack quack, doesn't say chick chick, it says quack chick. What is important is the LCMS is successful now, and that is why pharmaceutical research has really flourished because of the development of LCMS. Okay, so we get into the mass analyzers quickly. Okay, so there are several mass analyzers. I am going to talk only about quadrupole mass analyzer and maybe on time of flight mass analyzer quickly. So quadrupole mass analyzer is mostly used kind of a mass analyzer. So 90% of the mass spectrometers which are used either in LCMS or GCMS, they use a quadrupole mass analyzer. So as you can see, it has 4 rods, and we apply a DC and RF voltage in such a way that as we scan DC RF voltage, one ion gets stabilized and reach the detector at one particular DC RF voltage. But at that DC RF voltage, all other ions go out. At some other DC RF voltage, the ion which is showed by red color will go to the detector, but the ion which is shown by green color will go out. So by scanning DC RF voltage, we can make each and every ion to reach the detector one after the other. So in a scan mode, we can measure all the ions in a given sample, whether it is fragmented or not fragmented. We will be able to scan them and can get a mass spectra. Now, quadrupole has one more advantage, that if I want to do a quantitative analysis, and I know which ion I want to quantitate, I can select DCRF voltage in such a way that I will only allow that ion to go to the detector all the time, because now I'm not scanning DCRF voltage. So at that DCRF voltage, only one ion goes to the detector, and I can measure it 100% of time, so I can get maximum intensity. This mode is called a single ion monitoring mode, SIM mode. So the quadrupole can be operated in the scan mode, which gives us qualitative analysis. And when you operate quadrupole in the SIM mode, it gives us the quantitative analysis, because SIM mode is the most sensitive mode, where you are selectively monitoring one single ion of your interest. Okay, so it can be operated in this mode, these two modes, and especially this SIM mode makes it really very useful in quantitative analysis, whether it is metabolite identification, or when you do any kind of a BAB studies, or whether it is impurity profile analysis, any such analysis. When quantitative analysis is required, quadrupole is always preferred because of this SIM mode of its operation. Time of flight is very useful as it gives you high resolution. It can give you the molecular weight information up to 4th decimal level, and that gives it some specificity, because when you are working with some unknown metabolites, unknown kind of impurities, if you get a molecular weight information up to 4th decimal level, it definitely gives you added advantage that you can come closer to the structure of the compound which you are measuring. So time of flight is a very good mass analyzer, which gives you high mass accuracy. Okay, I'm again not going into that much. And you have a time of flight with Electron, which has still better specificity in the mass accuracy level measurement. And most of the time of flight mass analyzers are used today with Reflectron. Okay. So let us look at why we require LC-MS. Okay. So in case of a Gcl, you get a lot of fragments. While when you do analysis by LC-MS, you get a mass spectra like this. So I'm using only LC-MS. This is 100 nanogram of salbutamol, and I'm only getting a molecular ion. 239 is the molecular weight. Generally, a proton is added in electrospray ionization, and you get a molecular ion of 240. You do not see any fragments. So if this molecule is not known to me, and if I get a spectra like that, I will only know the molecular weight. If I want to know about this in more details, I need to fragment it. And fragmentation is only possible in case of LC-MS by using what is called as Msms technique. So we use Msms technique. So you have the 3 quadrupoles which you have to use. This is the central quadrupole, which is called as collision-induced dissociation cell. On one side, you have one quadrupole. You have another quadrupole on this side. So what you do, you select the ion, say this is salbutamol molecule with mass 240. Okay, that comes into the collision cell. When it comes here, I give a RF voltage to this quadrupole rods in such a way that this ion starts vibrating here very vigorously. From the other side, I introduce a leak of argon gas, and that argon gas molecules collide with this vibrating 240 ion of salbutamol, and it breaks it down, and the dissociation of this 240 ion will create the fragmented ions which are driven out into the next quadrupole. This quadrupole will work in a scan mode and will give us the information about what are the fragments of 240, and that will give us the structural information about this particular ion. Okay. So that is how it works. Okay, so it is called as collision-induced dissociation cell. So what actually is done is, suppose I have a molecular ion of mass 420, it comes here, it gets fragmented because of the collision, and the fragments go out to the next quadrupole, which will reveal the structural information about that. So what is done is 420 को अंदर लिया, उसको दिया punches, then it breaks down and reveals the information about what it has done, and that is how the crime is invented. And CID cell really works like a CID cell. Collision-induced dissociation is working like a CID. Okay, so this is how hardware flow is. So you have an electrospray ion source, you have the first quadrupole collision cell, and the next quadrupole. So this is how the triple quadrupole LC-MS system looks like. Okay, fine. So actually what happens then? So let us look at the situation. This is first quadrupole. There is a collision cell with a quadrupole here. This is third quadrupole, which is actually working as a mass analyzer. So now how we go ahead? We select only one ion in the first quadrupole. So in quadrupole 1 operates in SIM mode. So it will only allow one ion to get in. So all other ions have gone, one ion remains. It comes to the collision cell, gets fragmented, and the fragmented ions go to third quadrupole. And if I want to know more about this particular ion, I measure all the molecular weights of all these ions, and I can know how this molecule is made up of. If I want to quantitate it, I allow only one ion to go to the detector, okay, and then I measure that. And in this mode, when I operate the mass analyzer, it is called as the quantitative analysis of a drug in what is called as MRM mode, multiple reaction monitoring mode. So what I'm doing, the ion which I have selected here is called as precursor ion. It is fragmented to give me a product ion. And when I monitor this reaction of precursor ion giving me this product ion, then I'm using this for quantitative analysis. And especially when we are doing analysis of drug in plasma, we use this kind of a technique, especially any drug in biological matrix is difficult to analyze. So when you have this kind of a transition, you have more confidence in doing a proper quantitative analysis, because the mass, say salbutamol of 240, there could be many other compounds in your biological matrix which also could have 240 mass. They may be peptides, carbohydrates, or anything which also will get ionized in electrospray and will give a mass of 240. So if you use only single quadrupole, you are measuring 240 because of your molecule. You are also measuring 240 because of interfering chemicals. But when you measure this transition, this is very specific to salbutamol. You may not have a peptide which again give you the same fragment which is given by salbutamol. Okay, so that is how the specificity comes, and you can get a better quantitation by using Msms technique. Okay? So you can get a chromatogram for this particular transition. Area under the curve is proportional to the concentration of this in a drug, and that is how you quantitate it. Okay? Well, that is there is again a hybrid QTOF kind of a mass analyzer which gives you accurate mass because of time of flight. So there is a hybrid mass analyzer like quadrupole connected to TOF. Okay. Well, these are all the applications of LCMS in pharmaceutical industry: drug discovery, metabolism, which I'm going to talk a little bit about, toxicology studies, okay, quantitation, then contract research organizations, CROs, which do a lot of preclinical and clinical trials, and generic drug companies use it for impurity profile analysis. Biotechnology, I think somebody has already covered about proteomics and application of biopharmaceuticals, and LCMS or MALDI kind of equipment for biopharmaceutical analysis. Nucleotide and carbohydrate, I'm not getting into more. Forensic science, forensic toxicology is again one of the fields where mass spectrometry is very useful. LCMS as well as GCMS are very useful. In clinical labs, neonatal screening, therapeutic drug monitoring, drugs of abuse, and now LCMS is also entered into the diagnostic labs. So a lot of diagnostic labs like ThyroCare and the Sarabhai and what else, Metropolis, all of them have multiple LCMS systems. Okay, LCMS is also used in academia. Okay, fine. So let us now go to the next presentation. As I said, I have presentation, couple of them, and I will go to metabolite identification quickly. Okay, so we'll talk a little bit about metabolite identification, and that will, yeah. It's a huge presentation, but I will cut it short because of the time, and I will cover some of the aspects. Okay, so I hope it is visible to you. Yeah, you are able to see the slide?
Yes, sir.
Yeah, okay. So let us talk about drug metabolite identification. Okay, so introduction to drug discovery. We know this process of ADME and the drug metabolites. It is a vital however costly and time consuming in the drug discovery process. To identify metabolites is most difficult and most essential kind of a process. Okay. So where does metabolism take place? It takes place everywhere, in animals, plants, water, soil, everywhere, and liver, lungs, spleen, gut, various cells. So we have two ways of formation of metabolites, in vitro and in vivo. Okay. Fine. So when we have a metabolite formed, I mean, when we would like to really study metabolites of a drug, a new drug or new chemical entity, just to make sure that the metabolites are safe, they are not toxic, they have the therapeutic value or not, etc., then we need to have a method of analysis. So probably this slide itself will take a lot of time, but then as we have covered LCMS, it doesn't matter if I do not cover rest of them. Okay. So collection of biological, animal or human samples after drug administration or incubation of drug with liver slices, cells, microsomes or recombinant enzymes. Okay, so we can use any of the methods, in vitro or in vivo, to form the metabolites of the drug. Do some kind of a purification of them. I will just again put my then do some kind of a purification by filtration or by centrifugation or by even solid phase extraction, etc., like this kind of a techniques. And then initially you do the detection or identification of them, and then we do the characterization. Okay, so how the detection can be done? So these are detection of radioactively labeled metabolites by LC using radioactivity detector. So there are detectors available like UV detector, which can detect the radioactivity. So you use a label drug which has either C14 or tritium loaded on them, and because of that, the drug will have some radioactivity. And if you use it in vitro and try to get the metabolites extracted from the biological matrix and put them onto this LC with radioactive detector. Now this detector only will sense the radioactive compound. So if you separate them on a column, you will be able to identify the compounds which show you the radioactivity. So the peaks which show radioactivity are the compounds which are the drug getting converted into its metabolites. Okay, so that is how you can measure the retention time of those compounds. And then when you use any other technique, so this at least will be able to tell you that how many metabolites are getting formed in a particular process of in vitro or in vivo metabolism. In vivo, you have to take special ethical committees permission, etc., if you are going to use any radioactive compound onto any animal. Okay. Detection of new peaks in LCMS, total ion chromatogram and LC-UV chromatogram, comparison to the chromatogram of the blank. Okay, so when you see some new peaks getting formed when you use LCMS or LC-UV chromatogram, and those peaks could be the peaks because of the metabolites. Okay, so compared to the blank. So when you run a blank, you will be able to see hardly anything, but when you run your metabolite extracted sample, you will be able to see a lot of peaks, and those extra peaks compared to the drug by itself, you will be able to, or the blank plasma by itself, you will be able to identify the metabolites by that way. Detection of expected metabolites in ion chromatogram with drug with +16, -14, +176, etc. So when the drug metabolite gets formed, there will be either a gain or a loss of weight because of some addition of a group or some removal of a group from the drug structure. Okay, so +16 may be the oxidation, epoxide formation or oxidation has happened, because of which the molecular weight has increased by 16. -14 could be because of loss of methyl group. You have 14 loss, plus proton gets added at that place, so because of that the loss is not 15 but 14. 176 also because of gluconization or something, you will have addition of 176. we'll see what happens there. Detection of metabolites in ion chromatogram using stable isotopes, labeling of if available. So instead of using the C14 or tritium kind of radioactive isotopes, you can use C13, which is a stable carbon isotope, or deuterium instead of tritium. And if you substitute in a drug structure these isotopes and use mass spectrometer to identify the change in weight of these isotopes, you will be able to identify the metabolites on the basis of the substitution of carbon-13 or substitution of deuterium in the actual drug structure. Okay, so that can identify the metabolites. Detection of possible metabolites by neutral loss and precursor ion scans and using specific marker ions. Okay, so this is also possible. Unfortunately I have no time to talk more about precursor ion and neutral loss scan and marker ions, etc. So, but understand that there are some methods by which one can identify metabolites by using some of these scan events. Detection of possible metabolites by LC high resolution TOF-MS. Okay, so this also helps a lot because time of flight mass analysis gives you molecular weight up to 4th decimal level. So you can also visualize what kind of metabolites get formed and what could be its molecular weight. So you know the molecular weight of that metabolite up to 4th decimal level, expected metabolites. And then when you run your LCMS, if you see that molecular weight shown by your high resolution mass spectrometer up to 4th decimal level with accuracy level of few ppm, you can identify the metabolite on the basis of that. After that, you are going to characterize that. You have to find out the structure of metabolite. So comparison of product ion spectra of a metabolite and the parent drug. Okay, so the parent drug fragments in the collision cell and you get certain product ions of that particular original drug. Now the metabolite also has similar structure. So when you fragment that metabolite, you may get product ions which have some similarity with the product ions of drug. The fragmented ions of drug and the fragmented ions of metabolite will have some similarity. Some molecular weights will be same. And if you find that kind of a similarity, then you can say that this could be a drug metabolite because it shows similar kind of a trend given by the drug. Okay, so MS or MSMS is very useful. MSN kind of ion trap is very useful for that. Determination of elemental composition by accurate mass measurement. So when you do the accurate mass measurement, you get up to 4th decimal level molecular weight, and that gives you a proper elemental analysis, like how many carbons are there, how many hydrogens are there, how many nitrogens are there, how many halogens are there in the compound. And on the basis of that, you can visualize that yes, when this kind of a structure is there, so many carbons, so many hydrogens, etc., then probably this could be a metabolite. Okay, then components by MS analysis, for example, complementary MS analysis, for example, reverse polarity, different ionization methods, etc. So you can use, if you are using electrospray and atmospheric pressure, chemical ionization also is possible to be giving you similar kind of information. So you can use ESI and APCI both and look at the fragmentation pattern in both the modes and compare them, and if you get similar kind of information for the particular peak which is supposed to be a metabolite, you can on the basis of the product ions you can conclude, yeah, it is definitely the same compound which you are looking for. Synthesis of the presumed metabolites and comparison of the retention time and the spectra. Okay, some of the metabolites can be synthesized in the lab. Okay, so you can synthesize those metabolites and then compare the MSMS spectra of the synthesized metabolite and the metabolite formed in vitro or in vivo studies, and if they match, you have a confirmation. Then LC NMR, MS or offline NMR analysis after production and purification of the metabolite in a large scale. So you can also use other parallel technique like NMR, provided you have collected some small quantity of the metabolite in a process by using some kind of a preparative liquid chromatography or something, and then analyzing that compound by NMR for confirmation of the structure of metabolite. Okay, so this way you can find out the metabolites present. Okay, I will quickly go through what is required. Many metabolites can be predicted by using mass spectrometry. Processing is time consuming and repetitive, need automated processing. So this is where the research is going on now. Not all metabolites can be predicted, need to search data for unknowns. They may be hidden by abundant endogenous peaks, especially a lot of biological matrix. Data processing is extremely labor intensive, need automated processing of data. So the research is going on in this direction, or whatever has happened when you have various softwares available with various manufacturers for metabolite identification, and those can process the data very fast, and it reduces the drug discovery process because of that. May need to acquire Msms data, set up processing and reporting in is time consuming, need automated method setup and processing. So instrumentation is, again, this is processing of the sample, processing of the data, and this is the instrument manufacturing automation in such a way that you can set up the process of fragmentation, process of analysis of those metabolites, the fragments, etc., is done fully automatically. Okay. Yeah, so everybody knows that there are two types of metabolism, phase 1 and phase 2, and generally when you are working with metabolites, prerequisites for identification of metabolites: knowledge of organic chemistry is essential, knowledge of drug metabolism and basic metabolic reactions is necessary, knowledge of concepts of MS are required, interpretation of MS spectra for structural elucidation is required, and then if NMR is going to be used for confirmation, interpretation of NMR spectra for structural elucidation is required. So for metabolite identification, you require all these kind of knowledge base, so that the analysis of metabolites become very easy. Okay. These are some of the basic needs. I'm not going into that. So there are various instruments which can be used for that, which I already know. I'm just going into some aspects of some work which was done by one of my students. I will just check. I have the presentation on that, but again, I'm not going into the entire presentation. I will see if I can show you a couple of slides indicating what we really did. Okay, so this is the work which was carried out by one of my students on drug metabolites. Okay, so what compounds we use, etc., I will show you that. Okay, where it is. Yeah. Okay. So we worked on two drugs. One is amlodipine and other is etodolac. Okay. Yeah. So these are the two drugs. I will just come back and quickly go through that. Yeah. So etodolac and amlodipine both are chiral drugs, and the purpose of using chiral drugs for metabolite analysis was to see whether any kind of inversion happens. So when the metabolite gets formed, whether the chirality changes. So from R form, whether partially it becomes S form, or S form gets converted to R form or not. So that was the idea of looking at the metabolites of these two drugs, and we did some work on them. So we separation of HPLC was used, forced degradation studies we used, isolation and identification of degradation. Then we did in vitro metabolic studies, determination of chirality of chiral inversions, etc. And we also tried to find out in silico toxicity studies of the metabolites which we have identified. Okay, so I'm not going into the entire presentation, which was her PhD presentation for the Viva. So the part 1 I'm eliminating. I will go directly to part 2 to just show you how we did the analysis of the metabolites. Okay. So part 2, in vitro metabolic studies of those two drugs. So we had a method of extraction. We followed this. We took the liver cells and then worked with liver cells. So in vitro experiment using human hepatic cell lines. and work with that. So this is one way of preparing metabolites or making metabolites available by using in vitro studies. So this is all the sample preparation technique. Finally, we used LC-MS for the analysis. We use protocol for in vitro by using chicken liver tissues. Okay, so we use another method for metabolite formation. So we develop metabolites by these two different methods for both the drugs and analyze the metabolites by this. And I'm not going into, so these are the analysis of the metabolites. Okay, so various metabolites have been identified. So you can see we have identified 6 metabolites in this is etodolac, and we analyzed several other metabolites. These are etodolac metabolites. Okay, 6 metabolites have been identified. Their structures have been identified. I'm just quickly going through the kind of work which was done. Then we did similar work on amlodipine. We also found out the in silico toxicity studies also. So I'm not going into that data. This is amlodipine. So what is mobile phase used for analysis of metabolites, etc. is here. We got metabolites in various modes, and we found, I will show you, we found 12 metabolites for amlodipine, and many of these metabolites were reported for the first time. Okay, so, and we found out the structure of these metabolites. These are all product ions which are formed, and from the product ions we could find out the structure of all these 12 metabolites. Okay, and we of course used, for wherever it was possible, we used for confirmation NMR. So these are structures of various metabolites. So M1, M2, M3, M4, M5, M6, M7, M, like that. Okay. So this is chirality inversion studies. So you can see, instead of getting the peaks in the same area ratio, because we started with the racemic mixture of drug, and we could see that there is some kind of inversion happening in case of some 2 metabolites. Okay. Yeah, these are all the toxicity studies of metabolites. This is conclusion. Okay, so fine. So I just wanted to show you some applications also, instead of just covering the topic on mass spectrometry. So I think though it was not really very well arranged kind of a lecture, I thought probably I have given you some insight into some aspects of mass spectrometry and its application into the field of metabolites. Okay, so thank you very much for giving me this opportunity, and I hope it was probably acceptable to you the way I presented. Generally, I would be more arranged in giving any kind of presentation, but today I was not. So sorry for that if it has put you in some kind of inconvenience. And thank you for this kind of opportunity to talk to you. Any questions if you have, I will take a few of them.
Instead, sir, we are very thankful to you that only because of you and Dr. Amrapurkar madam, today our faculty development program, we managed to conduct it smoothly. Dr. Amrapurkar madam is there also with us. I will request her to express our feelings to
Yes,
Dr. Datar.
Thank you, Mrunal. Sir, a big thanks to you for cooperating with us.
My pleasure, ma'am.
Yes, sir, thanks a lot. Now you have given a very strong message that for the identification of the metabolite, we need to have the collaboration with the chemist, with the pharmacologist, analyst, and so on.
Yes.
So
Yes, absolutely
we have
true.
to go for the collaborative studies. We do not have any option.
Yeah.
Because this is all, very specific work and which really requires expertise in the field.
Yeah.
So
true.
very thanks a lot, sir, for a very nice lecture and with a very nice slides with your cartoons. Excellent, sir. Thanks a lot, sir.
My pleasure, ma'am. Yeah. If anybody has any question, I can take a couple of questions.
Yes, sir. First, Dr. Mandar will just sum up the session and then we will go for the questions. Is it okay, sir? He will
Yeah, it's
shortly
okay.
sum up the session.
Yeah, yes. Quickly. Yeah.
Thank you, ma'am. Yeah, thank you, ma'am. Thank you, sir, for giving such an enlightened session onto the LCMS applications for the pharmaceutical products and their metabolites. I hope all of us have enjoyed it very well. And what the way you have explained it with the normal common situations, the concept of CID and then the different application parts of it was very really nice. The metabolites identification process we will discuss in detail the way you describe the entire procedure and the significance of the procedure in each step was the really a good takeover from this presentation. The main highlighted part of the presentation, I will say that was the LCMS used in the chiral compound metabolites identification. That was the really good work what you should have done, and we really got to know from it very well that how the LCMS will be the most useful tool for the metabolite identification. So thanks for that, and I really from the KMK family, we all thanks to accepting our request in a very short notice period and then did a very wonderful presentation for us. Thanks a lot, sir.
My pleasure. Thank you.
The session is open for question answer. If there are any questions, I will request Melroy sir and Mandar sir to please take up it quickly because
Yes, ma'am.
sir
Please.
is having a next assignment.
Yes.
Yes, ma'am.
Session is open for the question answer. Most of the people are writing thank you to you, sir, for giving such a wonderful session.
Yeah.
Requesting participants to post your questions.
Sir, I have one question
Yeah, please.
regarding the whenever we go for the LCMS, the main thing problem with any compound or any method is we transfer from RP, that is the reverse phase, to the LCMS. So many times the equivalence between the mobile phase doesn't take place.
Yeah.
So how to approach in such situation, that I want to know.
Yeah, especially the method transfer from LC to LCMS is generally very simple unless and until you are using inorganic buffers like potassium, hydrogen, phosphate, etc. Because inorganic salts or those kind of buffers are not acceptable for MS. Okay, so there only you need to change the method. Otherwise, if you are using any kind of organic buffers like ammonium acetate, ammonium formate, etc., there is no need to change the method. You can just go ahead with the same method. Only provided, I mean, if you want to use a smaller flow rates and small column, especially the what we call as the UPLC columns. If you want to change from normal columns to UPLC column, you need to definitely change the method. Okay, because that may be better to give you higher sensitivity in MS if you use a sub 2 micron or sub 3 micron column. You will get better sensitivity, and in that case you need to change the method. But unless and until you are, I mean, if you are using the organic buffers, there is no need to change the method.
Okay. Okay. Thank you, sir. Thank you.
Yeah. Yeah. Any other question?
Request our participants to ask the questions.
Yeah. If anybody has any question?
Yeah, there is a question
Yeah.
from our own faculty, Dr. Pandana. She is asking that is it a challenge to use this method for herbals?
No, not at all. In fact, many of my students have used it for herbal products, especially phytoconstituents. And in fact, more amount of work, at least, I mean, I have guided some 10 students so far, and most of them, 8 of them worked on the phytoconstituents, and we used LCMS to find out the structure of the compounds which we could identify or separate from the phytoconstituent extract, and could identify the antimicrobial activities, etc., of those compounds. I mean, we not just determine the activity of the extract, but we could also identify which compound is responsible for that activity, and we also could find out the structure of that compound by using LCMS. So yeah, there is one method which I used was autobiography kind of a technique, which was very useful kind of a technique.
Just an extension to that question.
Yeah, sorry.
Yes, just an extension to that question. Is there any kind of library available for the plant or herbal metabolites?
No, there are database available, because as I said, there is no library available, direct library available as it is available for GCMS, but there is a lot of database available, and using that database you can get some idea about what kind of a compound could be present. And then you have to use many a times. I mean, when I did the work, I had to use other techniques like NMR and IR for confirmation of a structure. So not that LCMS alone is capable of giving this information. You have to use some parallel techniques like complementary techniques like IR or NMR for confirmation. Okay, so phytoconstituents, especially, I could collect more amount of sample and I could use that technique very well, those techniques very well.
Okay, thank you, sir. Thank you for answering that question. Surprisingly, I think there's
Yeah,
no.
Mandar,
Yes, ma'am.
can I have one question?
Yeah, please, ma'am.
Yeah, please, ma'am.
Yeah, sir, can you please suggest something for the prodrugs? So
Sorry?
what about the prodrug concept?
Mm-hmm.
So if the, I mean, we can have the active metabolite.
Mm-hmm.
And so in the research, how do we go about it using LCMS?
Yeah, especially, if you can find out the structure of the active metabolite, I mean, if it has a more therapeutic value than the actual drug, and then if it is stable and it can be synthesized in the lab, okay, knowing the structure, one can easily develop a method for synthesis of that. Only thing which needs to be studied is its stability and the way, whether it is really economical to synthesize that, and whether it can be easily synthesized or not. But yes, it is possible to have active metabolite. I mean, that can in fact give a clue to a new chemical entity.
Okay, thank you, sir.
Yeah, so that is possible. Yes, any other thing? It's not?
No,
Yeah.
sir. Thank you.
Okay,
Yeah,
thank you so much. And then I think I can leave.
Mandar, there is a question by Dr. Shrikant, sir. That excellent
Sir,
question,
we have
sir.
one more question
How
from
to
Dr.
make,
Shrikant.
yeah, how to make derivatization towards sensitive detection in HPLC, especially in case of aliphatic.
Okay, good. Okay, so probably I will take a leave because I have to go somewhere else. So thank you very much once again, and I will hang up. Okay, thank you so much. Yeah, bye bye.
Yeah, thank you, sir.
Thank you so much, sir.
Okay, since today our session has got over early, we will have a break time, and we all will rejoin at 11:15 for another very interesting session, because I know that every one of you have got now mentally exhausted after attending so many sessions for last 4 days, and therefore we are arranging a very interesting session on which Melroy will brief you on. I request Mr. Melroy to brief on the next session.
So we would now break for lunch. We would now break for tea. We have a very interesting session by Mr. Shreyansh Mehta from Art of Living, and sir is going to focus on a very light-hearted and a non-technical and a very soulful session. So I request all of you to join back sharp at 11:15, ma'am.
Yes, correct. And thank you, Dr. Mandar sir, for chairing the previous session. Thanks a lot.
Thank you, ma'am, for giving me the opportunity.
Thank you, sir.
Thank you.